Good morning, everyone. My name is Maria Victoria Melendres, and I'm here to present my undergraduate study in BS Biology at the University of the Philippines, Diliman. Crucial to the understanding of this study is the concept of cancer stem cell targeted therapy. So, say for example that you have a healthy garden, and then one day you notice unwanted weeds growing on one patch. So if you want to get rid of those unwanted weeds, it would make sense to target the source, which are the roots, rather than just removing the stems or the leaves so that the weeds don't come back and spread to other patches. This is the exact same logic that cancer stem cell targeted therapy is focused on, except the stems and the leaves are the heterogeneous cancer cells or the non stem cells, while the roots are the cancer stem cells. Our study focused on the effect of these plant leaf extracts, particularly the Garcinia rubra ethyl acetate and hexane fractions, on the MCF7 cancer stem cells. Garcinia rubra is a plant endemic to the Philippines, and a recent study has examined its therapeutic properties and assessed its cytotoxicity against three cancer cell lines, including MCF7. And what they found was that the ethyl acetate and hexane fractions had significant cytotoxicity against these cell lines. Cancer stem cells are essentially a very small subpopulation of cancer cells with stem-like properties, so they can give rise to the different cell phenotypes found in a tumor. They have enhanced proliferative potential, the capacity to self-renew indefinitely, and they are tumorigenic, meaning they can induce tumor formation when transplanted into appropriate hosts. Cancer stem cells are also unique in that they are resistant to traditional modes of treatment like chemotherapy and radiotherapy, which allows them to persist in tumors and cause cancer relapse. Since there are currently no established cancer stem cell lines yet, there was a need to isolate them from heterogeneous cell lines. And in the case of MCF7, those putative cancer stem cells can be isolated by culturing them as undifferentiated mammospheres, as shown in this photo, which are spherical clusters of cells containing cancer stem cells and progenitor cells. The discovery of the presence of cancer stem cells is relatively recent, as most cancer research has been focused on screening drugs against heterogeneous cancer cell populations, not necessarily accounting for the behavior of the diverse cell phenotypes when treated with a drug. One study has even suggested that it is possible for a drug that is seemingly non toxic to heterogeneous cancer cells to be selectively toxic to cancer stem cells. And because of these findings that challenge a lot of what is currently known, it is becoming more relevant to explore the possible differential cytotoxicity of drugs. So our study aimed to analyze the effects of these extracts on the formation and development of mammospheres, as well as to quantify the cytotoxicity of these extracts. So this was primarily a preliminary study of the technique in isolating cancer stem cells, given our resources, and preliminary testing of the behavior of putative breast cancer stem cells when treated with natural products. So you may be wondering why study the formation and development separately. Well, as I have shown earlier, related literature has proven that a not natural product may have different effects on mammosphere formation and development. So, in order to meet our objectives, we first had to isolate putative breast cancer stem cells from the MCF7 cell line. So, the cell culture protocols are similar with maintaining monolayer cells, but with two important varying conditions. First, the medium had to be serum free, and second, it had to be cultured in suspension by plating them onto non adherent plates to promote the 3D configuration of the mammosphere. So we found that distinctly shaped mammospheres began to form at day 3 or at 72 hours, and succeeding days showed further growth and an overall increase in the size of the mammospheres. The next we treated the mammospheres either at day 0 and at day 4 to examine the effects of the treatment on the formation of the mammospheres and its further development. So upon treatment at day 0, we found that doxorubicin was unable to inhibit sphere formation even at the highest concentration, despite it being a well-known anti-cancer drug. So this is in agreement with existing literature that report the resistance of mammospheres to traditional chemotherapy. On the other hand, the Garcinia extracts were able to completely inhibit sphere formation at concentrations as low as 25 for EA and 12.5 micrograms per ml for the hexane extract. To confirm the trend we observed, we measured the area occupied by a representative sphere per concentration. While this gives us a general knowledge of the trend, we acknowledge certain limitations in measurement that would account for the inconsistencies and the lack of a well-defined trend, such as the 2D measurement of a 3D spheroid. So note that these graphs are only for quantitative confirmation of the observed behavior of putative cancer stem cells when treated with Yersinia rubra, ethyl acetate, and hexane extracts. Nevertheless, we can confirm here in figure A that spheres were still able to form when treated with doxo, and in figures B and C, the Yersinia extracts were able to inhibit sphere formation completely, especially at the highest concentrations. So upon treatment at day 4, the plant extracts were only able to partially dissociate the peripheral cells of the spheres at the highest concentration for EA and 12.5 micrograms per ml for hexane. Meanwhile, the doxa-treated spheres remained intact and were morphologically similar to the regular spheres. With these findings, we postulate that bioactive components in the extracts may have a role in inhibiting strong cell-to-cell binding or in disrupting existing bonds. Another possible explanation is that certain signaling pathways essential in sphere formation may be inhibited. However, due to the compact nature of the spheres, the treatment may be unable to completely penetrate the sphere, which is why there's only partial dissociation of the peripheral cells in the developed spheres. Lastly, we wanted to confirm if the extracts were indeed cytotoxic or the cells were only losing their stem cell-like properties. So the Resazurin reduction assay was done since the usual MTP assay was not applicable in this case as the spheres were in suspension. But the underlying principle is similar in that viable cells can metabolically reduce the blue Resazurin to the fluorescent pink resolution. So in this figure, the IC50 values of the treatments on mammospheres were higher than those on monolayer cells, revealing the cytotoxic effect of the Yersinia extracts. This may be attributed to the increased resistance of cancer stem cell enriched spheres, so a higher concentration is likely needed to selectively inhibit T cells. We can also observe in this graph and the previous figures that EA can selectively inhibit cancer stem cells as low as 25 micrograms per ml, which is below its known cytotoxicity, which was computed to be roughly 33 micrograms per ml. So in the case of the hexane extract, it was able to inhibit and dissociate spheres at a concentration similar to its known cytotoxicity, suggesting its promising potential as an effective source of treatment. Uh, yeah. Note also the drastic increase in cytotoxicity of doxorubicin, and while this had the lowest IC50 value, it was still unable to inhibit sphere formation unlike the plant extracts. So uh, though that method of quantification still needs to be further improved, the results of the study suggest that bioactive components in the Yersinia extracts may not only be cytotoxic to cancer cells, but also to putative breast cancer stem cells. And based on the results of our study, the EA extract is not as cytotoxic as hexane, but both extracts can inhibit sphere formation and cause partial dissociation of the developed spheres, highlighting two different mechanisms from the same plant. It is to be noted, though, that these are not yet definitive cancer stem cells, so further research may focus on determining the specific phenotypes and the composition of cells that comprise the mammosphere. But nevertheless, these findings reveal the promising potential of the Yersinia rubra extracts as effective sources of chemotherapeutic agents for cancer stem cell targeted therapy. And with this knowledge, we may be able to improve treatment regimens for cancer patients to be more efficient in targeting the source and ultimately preventing cancer recurrence. Thank you and good morning.